biotechnology revision, and then we'll do some important questions which are based on some concepts. Before that, if you want, you can tell me your doubts. It could be from any chapter, not an issue. Yes, so anyone has anything in mind? No doubts? Okay, no issue. So then let me go over with the biotechnology, and we'll first go through some important concepts which are whose weightage is more for this chapter, and there are more chances for these concepts to be asked in your board exams. Okay, and then we'll do some previous years NEET questions on these concepts, because clearly these concepts are important both for boards and entrance. Okay. Okay, so as we discussed in the class about biotechnological applications, I told you that it has a wide variety of applications, from IVF to vaccine development to gene synthesis or expression to genetically modified organisms, DNA fingerprinting. This all we studied in genetics, and gene therapy, right? And at the core of it, of everything that can be done through biotechnology, there are some core principles, which are two core techniques that lie at the heart of biotechnology. And what are these? Anyone would like to? Atif, what are these two core techniques that we studied, and also which are in front of your screen?
Yes, genetic engineering and bioprocess engineering.
Yeah, and what do you mean by genetic engineering and bioprocess engineering is?
Genetic engineering involves the techniques to modify chemical composition of genetic material, and it involves techniques to modify DNA and introduce it into host organism to study the phenotypes. And bioprocess engineering is the maintenance of a sterile ambience in the engineering process.
Yeah, right. So mostly, majorly, either we engineer the DNA, like we do changes to the DNA itself, right? If we are doing that, we call it genetic engineering. But there can be cases where we are not tweaking the DNA, but still is a biotechnological application, like the one you see here, IVF, right? What is
Yes.
IVF?
In vitro fertilization.
Sorry. Yes, what is IVF? In vitro fertilization. So what are we doing here? We are making the sperm and the ovum, which are the human gametes, fertilize with each other outside the human body, and then that in vitro. So it's called in vitro fertilization, and then that the embryo is implanted back into the human female's womb or uterus. Correct? That's what is done, right? When we do this, we are not tweaking, or we are not manipulating the genetics. We are not manipulating the gene of either of the gamete, but we heavily rely on the process. So this bioprocess engineering is where you do this technique of IVF in a laboratory condition, which is microbial free, microbial contamination free, okay, and is very high sophisticated, basically very sophisticated technique. Do you understand, everyone? So there is no, we cannot afford to have any contamination or anything that should go wrong in this process. Otherwise, the zygote or the embryo which is formed will be faulty and there will be issues with the development of that embryo. Okay, makes sense? So in this case, the second part, bioprocess engineering, becomes very important, right? Yeah, so this was one thing. And we also talked about recombinant DNA technology. So, and the tools. So this is a very important concept. What are the tools for recombinant DNA technology? So anyone would like to tell? Donna, would you like to tell me what are restriction enzymes, which is a tool for recombinant DNA technology? You're there, Donna?
Yes, sir.
Yeah.
Uh,
Remember we talked about restriction enzymes?
kind of.
Yeah.
I think it is a protein produced by the bacteria.
Mhm. So these are proteins, you are right.
And yeah.
Enzymes are mostly proteins, and they were figured out in bacteria. And what do they do? Why are they
It
called restriction enzymes?
it cuts DNA at specific sites.
Yes, it cuts DNA. So there are two types, right? Exo and endo. What does,
Yeah.
yeah, what does this mean?
If I'm not wrong, I think endo restriction enzyme is the one that cuts at the ends of the DNA.
No, that's exo. So remember, I'm also showing you these things. So exo means outside. So from the sides, they chew the DNA. Okay.
Yes.
So they are not cutting at specific sites, they are cutting from the side. Whereas
And
endonuclease,
specific points.
yes, within, not from the ends, anywhere in the center.
Yes.
It can just cut here or here or here at specific site. Okay, depending on which restriction enzyme it is. Yeah. So what is more useful for biotechnological application, exonuclease or endonuclease?
Endonuclease.
And why is that?
Because I think we need to only cut like one side to insert the foreign DNA. We don't need to cut both the ends.
Yeah, so basically what we need to do is we need to insert or we need to take out any gene or make any gene inactive, right? Put new gene or make the gene inactive. These are two things that we do, and for both, cutting at a specific site is very important.
Yes.
Yeah, good. And we talked about some rules for naming these endonucleases. Do you remember this? I explained it through two examples, EcoR1 and HIND2.
Yes.
Yeah.
The rules were that the first 3 letters is the source organism, and the 4th letter would be the strain,
Right.
and the number is the order of isolation.
Correct. So the first 3 letters that you said, in that also, the first one is the genus, like in ECO, EcoR1. E is genus Escherichia and CO is for coli, right?
Yes, sir.
Cool, good. And I also quickly, I told you that what is the role of palindromic sequence? What is the importance of palindromic sequence in this whole cutting through restriction enzyme business? Do you remember any importance of it?
I'm not sure about this, but is it that no matter where you cut in the no matter where you cut, the order is going to be the same, something like that?
No, so palindromic sequence, you are very close, but palindromic sequence is not about where you cut, it's about no matter which orientation you read it. So the sequence is same from both. So if you go here, like there are two strands of DNA, right? Running from 5' to 3' and the 3' to 5'. So if you read it this way, it is GAATTC on one strand. On the other strand, if you read it again this way, opposite way, 5' to 3', it is again GAATTC.
Yes.
If you read it the other way, 3' to 5', CTTAAG. Here also CTTAAG, right?
Yes.
So these kind of sequences are called palindromic sequences. There are palindromic words as well, like Malayalam is a palindromic word. No matter from which direction you read it, the same alphabet will come in the same order, right?
Yes.
Right. So, and the important thing is that these restriction endonuclease, if you have not written it, you can just note it down that these restriction endonucleases, they recognize one or the other palindromic sequence where it creates a cut. Now the question is why to choose a palindromic sequence to create a cut? What is the benefit of choosing a palindromic sequence to create a cut? The answer lies here. Can you explain this? Hanifa. Hanifa, can you explain what's happening here? What did we discuss? Are you there, Hanifa? Or Heba? Heba, are you there?
Yes, sir.
Yeah. So what is happening here about why restriction endonucleases choose palindromic sequence? Or they recognize palindromic sequences? Any idea, Hiba? Atif? I think we discussed this. Give it a shot, give it a try. What do you think? Atif, are you there? Okay, I don't think anyone wants to answer. So the reason that restriction endonucleases recognize palindromic sequences is that the way they create a cut is not exactly in the center of the sequence, but either towards left or towards the right of the center. And they always create a cut between the same base pairs on the two opposite strands. So let's say if they are creating a cut between A and A here, in the other strand also they will create a cut between A and A only. And this type of cutting leaves sticky ends. Okay. So sticky ends are very important to use this as a tool, because when you use restriction endonuclease and you cut a DNA, you can ligate it back, with other DNA fragment, and that's how we put our gene of interest back into the plasmid when we cut it through restriction endonuclease. Okay.
Yes, sir.
So that's why it's important, and that's how it does. Okay, anyway, so this was about restriction endonuclease, and it's an important concept, I think. Yes, the third important thing is about the cloning vector or the plasmid vector. Many questions comes from cloning vector, plasmid vector, and this question in general is important, that what are the characteristic features that are important or that are required to make a good cloning vector, or in other ways it can be asked, what are the characteristics that should be present in all cloning vectors. Okay, so there are three that has to be there, important ones: origin of replication, selectable marker, and a cloning site. Yes, so Atif, would you like to give this a quick revision? What does these three means? Am I audible, people?
Yes, sir.
Yeah, so I think no one is responding. Donna, would you like to quickly give
Sure,
it
sir.
a, yeah?
The first one, ORI, which is origin of replication.
Mhm.
It is responsible for initiating replication in DNA sequence, and also it controls the copy number of the linked DNA. For many copies of target DNA, it should be cloned in a vector whose origin supports like a high copy number.
Correct.
And um, another one point that I had to add to the first one, which is any piece of DNA when linked to the sequence can replicate within the host cells. Then we have the selectable marker, which
Mhm.
helps in identifying and eliminating the non-transformants.
Correct.
And transformation is defined as the procedure which a piece of DNA is introduced to the bacterial cell.
Yeah, right.
And genes encoding resistance to antibiotics like ampicillin and tetracycline are useful selectable markers for E. coli.
Correct.
And the also also one more point, the normal E. coli cells, I think, do not carry resistance against any of these antibiotics. I mean.
Right. Yes.
And
And
the third one is cloning sites.
mhm.
Cloning sites, it to link the alien DNA, the vectors require very few recognition sites for the restriction enzyme.
Mhm.
And I do not know any other point for cloning sites.
So cloning site is basically this. You can define it by two ways. One is through the restriction endonuclease. So the regions that these restriction endonuclease, that those palindromic sequence regions, right? We talked about.
Mm-hmm.
So the regions that these restriction endonuclease recognize and create a cut at, they are called the cloning sites. Because why are they called cloning sites? Because those are the regions which will be cut and where we will insert our cloning gene, the gene that we want to clone. Okay, so if I ask you where will your gene be present in that vector, so you will say simply where I can make a cut, right?
Yes.
Only there I can insert my gene. So that's why that region which restriction endonuclease can recognize and cleave are called cloning site, or in other words, the region where our cloning DNA is inserted are called the cloning sites in that plasmid. So these three has to be there. Okay, and that makes any cloning vector a good cloning vector, right? Yes, so that's there, that's important. And Then comes the process, and I think in process things are clear. So let me just go through it quickly. In a recombinant DNA technology process, what we do is we isolate the genetic material, and we talked about how to isolate DNA. And this is one of the practicals also, right, in biology that you have done in your schools, right? And finally, when we have the DNA, we cut it using the restriction endonuclease. And one important thing that you have to remember is to be careful to use the same restriction enzyme to cut our DNA from the genome, and use the same restriction enzyme to cut the plasmid vector as well, because they will cut at the same sites and they will also create same sticky ends, which are very important for ligating it back. Okay, so the same restriction endonuclease has to be used. Okay.
Yes, sir.
Then comes after cutting the DNA, we separate our DNA of interest, a small fragment. We amplify it through PCR, and we'll just go quickly through over PCR. So anyone who would like to just tell me quickly what is PCR this time? Atif or Hanifa? If you people can speak, that will be great, because it's me and Donna doing all the talking. So you're there? Yes, Hanifa. So what is PCR? Quickly, some important things that we should remember.
PCR stands for polymerase chain reaction. In this reaction, multiple copies of gene or the DNA is synthesized in vitro using two set of primers. There are three steps: denaturation, annealing, and extension.
Right, correct. And how do we basically achieve this amplification? What happens in these three steps? Am I, Hanifa, you're not audible if you're speaking. You're mute.
Denaturation, it is heated on 95 degrees Celsius.
Yeah.
To get two primers, and then then it's on 72 degrees Celsius, and then we get a DNA polymerase plus a tax, which is like a tax polymerase plus deoxynucleotides, and then in extensions they are extended, and like within 30 cycles we can obtain 1 billion copies.
Yes, correct. So you can just see that it's true that over 30 cycles you get, if you start with one DNA, you get lots of copies, around 1 billion copies. Okay, after we have amplified our gene of interest, we prepare recombinant DNA, okay, a vector. Then we use ligases and we insert our recombinant DNA, which we have just amplified here, into the host, okay, through the vector. And then we obtain the foreign gene product, because this is why we have done all this business, to put a gene in a host which can be a bacteria or a yeast, and then we grow and culture them in big vessels called bioreactors. And then finally we do the downstreaming processing. Okay, so in downstreaming processing, purification and separation are the two important aspects that we talked about, and it was also a question that I asked in your exam, one of the mock exams. So this is overall a broader quick revision, and let's spend now some time on questions. So before that, do you have any questions? Let me pull out some questions for you. Anyone has any question till here?
No.
No? Okay. take a 1-minute breather, and I will be sharing the screen with some previous year's questions in a moment. It's taking some longer time to open. Just a moment, people. Yeah, it's there. Okay, cool. So okay, yes, here it is, biotechnology. Okay. Cool. So can you all see the screen that I'm sharing? The questions?
No,
sir.
sir.
Oh, just why is that?
No, sir.
Yeah. Just 1 second. I'm doing it again. Yeah. Can you see it now?
Yes, sir.
Yeah. Okay. Very well. So we have the questions, and so I've selected some questions which have concepts that we can discuss along the way as you will be solving the questions. Okay, so let me so how we'll work is same. I'll select some questions. I'll ask you to read. So one of you will read, and then you all will send your answers in the chat, right? And any one of you, if you feel that you need a discussion on that question or you find it difficult to solve, so don't hesitate, write in the chat that you need a discussion. Okay, and I'll discuss the concepts behind the question and how to solve questions of similar kind. Rules are clear? Everything is clear?
Yes, sir.
Yes.
Okay, good. So let's start with a simple question and to test whether it's working, you all can send me answers quickly. So we'll start with question 4. So Hiba, would you like to read question 4? I think this is better, right? Can you read question 4? Hanifa, can you read? I think Hiba is not responding.
Genetic engineering is possible because we can cut DNA at specific sites where restriction in the nucleuses. Restriction in the nucleus purified from virus can be used in bacteria. The phenomena of transduction in bacteria is well understood. We can see DNA by electron microscope.
Yes, your answers, please. Okay, I'm getting your answers. So Atif has given the right answers. Atif has given the right answer. Atif, that's right. And I've not received answer yet from Hiba. Hiba, are you there? Hanifa and Donna, would you like to read that statement again that you have picked as a right answer? I'm sure that you will figure out what's wrong with that statement. Yes, so people, the right answer is option A. Yes, Donna, the right answer is option A, not B, because let's see why B, C, and D are wrong. B says restriction endonucleases purified from virus can be used in bacteria, but that's not true. They are purified from bacteria. They come from bacteria. That's why their name are EcoR1, Hin2, CLA1, etc. They are figured out from bacterial systems, not from virus. They are purified from bacteria. Is it clear?
Yes, sir.
Yeah. So B can't be correct. C says the phenomena of transduction in bacteria is well understood. First, it's not, and second, that's not how genetic engineering is done. It's transformation, not transduction. Okay, so transduction is different, transformation is different. D says we can see DNA by electron microscope. We cannot. We cannot see a single thread of DNA by electron microscope. What we can see is beads on string structure of a chromatin fiber at the best. So A, which says we can cut DNA at specific sites by restriction endonucleases, is the correct answer. And that is how we can genetically engineer the DNA. And that's how genetic engineering is possible, because you can cut DNA, put anything in or take anything out, whatever you want. Is it clear, everyone?
Yes.
Yes.
Yeah. Okay. So let's go to question number 11 quickly. Atif, would you like to read question number 11?
Yes, sir. Which of the following contains the key tools for recombinant DNA technology? Option 1, restriction endonucleases, ligases, vectors. Option 2, ligases, host organism, polymerase enzymes. Option 3, vectors, Taq polymerase primers. Option 4, restriction exonucleases, ligases, primers, bioreactors.
Yep.
So,
Your answers, please, in the chat. Yes, that's a correct answer, Donna. Yes, Atif, that's correct. Hanifa, also correct answer. Yes, Hiba, correct. So it's B, 1 and 2, which says we need which are the key tools? Restriction endonuclease, of course, ligases, vectors, host organism, polymerase. All of them are important key tools. Tag polymerase and primers are not that important as key tools because it's just for PCR. And again, bioreactors is for downstreaming processing, right? So option B is correct. Good. So finally, oh, I'm so sorry. Finally, a correct answer coming from all of you. Oh, I think I did something. Just give me one moment, people. Something went wrong. Okay, sorry, got shut down. I have to reopen it. It's a big file, it takes time to open. Yes, that's open now. Okay, can you see the screen, everyone?
yes.
Yes, sir.
Okay, great. Okay, so the next question that I have selected is question number 18. Can you read question 18, Donna?
Yes, sir. Read the given statements and select the correct option. Statement 1, restriction endonuclease enzymes recognize a specific palindromic nucleotide sequence in the DNA. Statement 2, restriction endonuclease enzymes are called as molecular scissors or biological scissors. Option A, both statement 1 and 2 are correct. B, statement 1 is correct but statement 2 is incorrect. C, statement 1 is incorrect but statement 2 is correct. And D, both statement 1 and 2 are incorrect.
Yeah. Your answers? Yes, so all three of you, Hiba. No, I think Hiba has not given an answer yet. So Atif and Donna has given the correct answer, and that also in good time. So that's good. That is the correct answer. Hiba, we're waiting for you. Hiba, are you there? Okay, I'm not sure if Hiba can hear me. So the correct, so let's discuss this. The correct answer is option A, which means both statements 1 and 2 are correct, right? So both are correct statements. Simple. Yeah, let's go to a little more conceptually complex problems. Yes. Question number 33. Donna. Oh, Donna just read. Atif. Question 33.
Yes, sir. Having become an expert on gel electrophoresis, you're asked to examine a gel for a colleague. Where would you find the smallest segments of DNA? Option A, near the positive electrode farthest away from the wells. Option B, near the negative electrode close to the wells. Option C, near the negative electrode farthest away from the wells. Option D, near the middle. They tend to slow down after the first few minutes.
Yeah. Your answers in the chat. Yes, Atif, that's correct answer. Yes, Hiba, that is correct. Donna, we are waiting for you, or do you
So
need
I
any
just sent
discussion?
an answer. I'll
You
send it
did?
again.
Yes, great. So that's correct. So I hope everyone understands the concept behind it, right? DNA being negatively charged will move towards positive charged, and the smaller the DNA fragment is, the faster it will move on the gel electrophoresis, right?
Yes.
Yeah. Okay, now we'll not go and do more questions on gel electrophoresis. That's simple, I think, for you. Yes. Now I want you to do question 38. Anyone? Donna, would you like to read question 38?
Yes.
So remember, don't get intimidated by the length of the question. It just stick to your basics. And yes, Donna.
Which of the following statements are correct? First statement: restriction enzymes cut the strand of DNA a little away from the center of the palindromic site, but between the same two bases on the opposite strands. 2. Hint2 always cuts DNA molecules at a particular point by recognizing a specific sequence of 6 base pairs. 3. Separated DNA fragments cannot be visualized without staining on an agarose gel electrophorus. And fourth one, ORI, which is the origin of replication, is the sequence responsible for controlling the copy number.
Yes, correct.
And fifth
Now,
one, there's one more.
yeah, read it.
DNA is a positively charged molecule.
Yeah, correct. So you have to see the options below and tell me which one is the correct option containing the correct statements. Yes, so Atif and Donna, correct options. Very good. And Hiba, yes, Hiba, would you like to read it again? So read your options. So your option contains the number of the statements that you think are correct. Yes, that's correct, Hiba. So I think that you realized what was the very simple and gross mistake, right? In option A. So fifth statement is straight out a wrong statement. DNA is not a positively charged molecule. DNA is a negatively charged molecule. So you just have to see. You have to do nothing. This question is actually very, very simple. If they're asking about correct statements, any option which will contain fifth statement will be a wrong option. And one A, C, and D contain fifth. Do you understand that? So outright A, C, and D cannot be the right option. Only option could be B. Okay, because every other statement apart from the fifth statement is correct. The fifth statement is completely incorrect. Make sense, everyone?
Yes, sir.
Yeah. Okay. Good. A similar kind of question is question 40. Atif, would you like to read question 40? Or Hiba, can you speak now? Would you like to participate and read the questions? Are you comfortable reading the questions, Hiba? I hope not, I think. Okay, Atif, over to you. Atif, can you read question 40? It's okay, Hiba. Hiba is not feeling very well today, but yeah, she's in the class and she's answering. That's good. Atif, are you there? Would you like to read question 40? I think if people have network issues or something. Okay, then
Sir, can you hear
going.
me?
Yes, I can hear you now, Atif.
Yes, sir. Question number 40.
Yes.
Okay, sir. Read the following statements and select the correct ones. Option 1, same kind of sticky answer produced in a DNA has been cut by different restriction enzymes. Option 2, exonucleases make cuts at specific positions within the DNA. Option 3, hint 2 was the first restriction endonuclease to be isolated. Option 4, a bacteriophage has the ability to replicate within bacterial cells by integrating its DNA with bacterial DNA. Option 5, a presence of more than one recognition sites for an enzyme within the vector complicates the gene cloning. Yeah.
Yeah, great. Quickly, your answers in the chat. Uh, yes, Donna, that's correct in option. Atif, would you like to just read again uh, the statements that you think are correct? Yes. Now that's correct answer, Atif. So, Hiba, we are waiting for your answer. Yes, perfect, Hiba. That's correct. Correct an option. So the correct option is C. Now, but I would like to listen from you people. C, which means 3 and 4. So statement 3, which is hint 2 or the first restriction endonuclease to be isolated, yes. And isolated means isolated from the bacteria and used. And option 4, a bacteriophage has the ability to replicate within bacterial cells by integrating its DNA with bacterial DNA. That's correct. Okay. Why are 1, 2, and 5 wrong? That's what I'm interested in. So, Donna, why do you think 1 is wrong?
So I eliminated the second one first because
Because
exonuclease usually removes nucleotides from the ends of the DNA.
yeah, this is exactly what we just discussed today, right?
Yes.
They chew the DNA from the ends. That's why they're called exo from outside. That's correct. So you removed second and you realize that but second is only present in option D, right?
Yes.
So how did you negated A and B? How is 1 also wrong and 5 also wrong?
When cut by the same restriction enzyme, the resultant DNA fragments have the same kind of stickiness produced,
Perfect.
and which can be joined together using the DNA ligase.
Yeah,
So I
so good.
eliminated the first option as well.
Yeah, and what is wrong with statement 5th? I think statement 5th is very close to be a correct statement. That's why I think Atif got confused in the first place, right, Atif?
Yes, sir.
So do you now understand what is wrong with statement 5 that you changed your answer to option C, which is the correct option?
No, sir. I'm not sure why it's wrong.
Okay, so you just change because I said so. Okay, Donna, would you like to? What is your opinion? Why it is wrong?
So I actually didn't check that. Like I didn't know the answer to that. just knew that the first one was wrong, so it couldn't be option A, and 5 is only in option A. So
That's another very smart way to filter out options. So this tells you one thing, that you don't have to know everything to score good in exam, right? You just have to know what is for sure wrong or what is for sure right. So statement 5 is wrong because they have not specified what enzyme. Presence of more than one recognition sites for an enzyme within the vector complicates the gene cloning. That's not always true, because if the enzyme that they are talking about is polymerase, that helps the cloning. If the enzyme is ligase, that again helps the cloning. But if that enzyme whose recognition site is more than one and that enzyme is restriction endonuclease, then it's a problem. So unless and until you, because restriction endonuclease is not the only enzyme playing a role in the process of gene cloning, correct? Do you understand, Atif and Donna?
Yes, sir.
So which enzyme? Yeah, so this is to trick you, because most of the time while studying NCERT, we always talk about the significance or importance of restriction enzyme. So we always say that there should not be more than one site, there should not be more than one cloning site, because if you create a cut at two places in the plasmid vector, the plasmid vector will become two linear pieces of DNA, which is useless to us. But here they have not mentioned what enzyme they're talking about. Okay, so there are many enzymes that play a role in the process. Ligase, polymerase, restriction endonuclease are among the few. Is it clear? That's why 5 is not the correct option.
yes, sir.
Yeah, okay, very good though. But I am glad that Arif changed the answer to the right and chose the correct option. So let me go quickly to the another segment. Yes, this one. Question number 62. It is a concept of DNA and the membrane together. Atif, would you like to read question 62?
Yes, sir.
Yeah.
DNA cannot pass through the cell membrane as option A, it is too big to cross the membrane. Option B, it is a hydrophilic molecule. Option C, membrane does not have specific proteins to facilitate the transport. Option D, none of these.
Yeah, and remember, the hint is in your biology practicals where you isolated DNA. So think about it and tell me your answer in the chat. Yes, Atif, that's correct option. Good, Donna, correct. Hiba, are you there? Do you find any difficulty? Hiba, are you there? Yes, finally. And that's correct answer, Hiba. So the right option is B. DNA cannot pass through a cell membrane as it is a hydrophilic molecule. Remember, that's why you cannot precipitate it out in water. So that's why you very carefully in the test tube where you are doing the genomic DNA extraction, you add what? Chilled
Chilled
ethanol.
ethanol.
Yes. So it gets it comes out in the chilled ethanol and then it precipitates right into in the form of thread like structures, and then you can spool it out using a very fine twig or needle or something, right? So it's hydrophilic, it loves water, so it gets solubilized in water. That's why the plasma membrane is hydrophobic, it is lipid based, so anything that can that is hydrophobic and can dissolve in lipid can only cross the membrane and not the hydrophilic molecules. Is it clear, everyone? Perfect, very good. So there's one more question which this is about the biolistics. Yes, processes in this one. So you have to put the steps in correct sequence. Let's just go through it. Question number 69, it is again what we discussed today. So Donna, would you like to read question 69?
Yes, sir.
Yeah.
Question 69. The different steps of recombinant DNA technology are given below randomly. First, isolation of the DNA fragments or genes to be cloned. Second, introduction of the recombinant DNA into a suitable cell, usually E. coli, called host. Transformation. 3. Multiplication / expression of the introduced gene in the host. Fourth one, selection of the transformed host cells and identification of the clone containing the desired gene / DNA fragment. Fifth one, insertion of the isolated gene in a suitable plasmid vector. Which of the following represents the correct sequence of steps? A, which is 1, 3, 2.
Yeah, don't read the options. So
Okay.
you have to figure out, okay, everyone. Option A, B, C, or D. Now, this is a little because you have to find the sequence now. So you have to. It's not like which is correct or not. So which is the what is the correct sequence? So your answers. Yes, Donna, that is correct option. Others, Atif. Yes, Atif, correct. That's correct option. Hiba? Perfect. So all three of you have chosen the correct option, which is option C. And let me give you a trick for these kind of questions when they come. So if you are sure about the beginning and the end of the process, then only look for that. Sometimes you will find more than one option. Then if you find more than, like in this case, one is the first step, isolation of DNA fragments or gene to be cloned. Okay, no step comes before it. And the last step of all the 5 steps that are mentioned is step 3, multiplication or expression of the introduced gene in the host. That is our final goal. If there was an option which talked about downstreaming process, that will be even later a goal than step 3. So that would be the last. But here 1 is the first and 3rd is the last. So without wasting any time and saving precious seconds, you first look which of the options starts with 1 and ends at 3. And it happened to be that there is only 1. So 3 options start with 1, but only 1 option ends with 3. So C is the right option. If there were 2 options starting with 1 and ending with 3, then you will have to go to the middle steps. Okay, there also you can go to the second step and see where it matches, and you can most of the times get the right answer there right away. Is it clear, everyone? So try to develop your instincts to save time in entrance while also staying on correct course to choose the right answer. Okay, Heba, Donna, and Atif.
Yes, sir.
Okay, we'll take a one last quick question from this today, and that is question number 80. This one, question 80. Atif, will you read the last question, question 80 for today? Atif, are you there?
Yes, sir.
Question 80. Yeah.
Okay. Which of the following statements are correct for the enzyme Taq polymerase? Option 1, it remains active during the high temperature induced denaturation of dsDNA. Number 2, it requires primers for carrying out the process of polymerization. Option 3, it synthesizes the RNA region between the primers using DNTPs and Mg2+.
Yes. Your answers, people. No, Atif, that's not the right option.